right, so we are going to dive into the chapter that talks about bacteria, protists, and fungi. And we're going to do this in 3 sections. That way the videos are a little bit shorter and it kind of puts the information into chunks. And so we're going to first start with our microbes and our bacteria. But before we get into some specifics about that, we need to look at some terms. Now, these are terms that if you took Biology 1, they should be review. If you've taken biology in high school or not too long ago, they should also be review. So let's take a look at some of this with the terms. So the first thing we want to look at term-wise is prokaryotic versus eukaryotic. So prokaryotic cells are cells that have a membrane around them. They have very simple structures in the sense that they have DNA located inside, and they have ribosomes. They do not contain a nucleus, and they do not contain all these little organelles. Now, organelles are little organs that do specific jobs. They don't have those. And so these are pretty simple cells, okay? So they don't have a nucleus, and they do not have membrane-bound organelles. But they do have ribosomes, and so they're a simpler cell. And you can see them located here. Now, eukaryotic cells, on the other hand, they actually have a true nucleus inside. Okay, this nucleus is where the DNA is located. It's got an extra layer of protection. So not only does it have the plasma membrane around the cell, it also has a nuclear membrane around the DNA inside the cell for an added layer of protection. Another thing we notice too is that it has organelles present. It's all these tiny little structures inside the cell that do specific jobs. Like the mitochondria is the powerhouse of the cell. Lysosomes are going to clean up the cell. They're going to recycle things. The rough ER is going to help make proteins. Smooth ER is going to help make lipids or fats. So they have special jobs. This means these cells are more complex. Okay, so prokaryotic, simple, do not have a nucleus. Eukaryotic, more complex, does have a nucleus. Okay, so you can see the pictures located here. Some other terms we want to look at is what we call unicellular versus multicellular. Unicellular means it's made up of one cell because uni means one. Okay, so like unicycle has one wheel. So in this case, they have one cell. Other organisms are going to be multicellular, meaning they're composed of many cells. Okay, you are multicellular. You're composed of many cells. All right, so now let's talk about nutrition. How do these cells and these organisms gain their nutrition? So there's 2 main groups that fall into this. There are autotrophs and there are heterotrophs. Autotrophs are going to make their own food. Again, breaking down the word, auto means self, troph means feeder. They are going to feed themselves, and they can do this 2 ways. They can do it through photosynthesis, like plants can, using the sunlight in order to make their own food. Or they can do what we call chemosynthesis, where they use chemicals from the environment to make their own food. This would be like bacteria that lives at the bottom of the ocean where sunlight doesn't reach. Okay, they still can make their own food, but they're going to do it differently because there's no light down there. Photosynthesis requires light. On the other hand, heterotrophs are going to have to rely on their environment for food. They're going to have to actually consume their food or eat their food in some type of way. When we talk about heterotrophs, they could do this by absorbing their nutrients. This is what decomposers do, like fungus. They liquefy their food and they suck it up, kind of like a sponge. This is what happens whenever stuff starts to mold in your fridge, how it liquefies. And it's because the mold is a fungus and it's going to then absorb its nutrients, its food. We are heterotrophs as well, but we do ingestion. We're an ingester, meaning we have to take the food in and then digest it. All right, so we take the food into our mouth, it then goes into our digestive system, and that's how we get our nutrients. Okay. And so when we look at autotrophs, autotrophs a lot of times are known as producers because they produce their own food, whereas heterotrophs are known as consumers because they have to consume or eat their food. The last set of terms I want to talk to you about are sexual versus asexual. Now, this is referring to reproduction. So when we're talking about cells and organisms, we need to look at the two forms of reproduction. And the first one here is sexual reproduction. Sexual reproduction involves the process of meiosis and fertilization. So let's talk about what that means. Meiosis means that we are going to see the formation of egg and sperm. Okay, so we're going to have male and female, and they're going to make egg and sperm. Those egg and sperm then have to come together and fuse in fertilization to make a new organism. The purpose of this is to create offspring that are genetically different. Every individual is going to have a difference in its genetics. So this is why you may have siblings from the same parents, but you look a little bit different. Some things may be kind of the same, but others not. And it's because the egg and sperm had unique characteristics for you versus your brother or your sister. So sexual reproduction gives us diversity, okay? It allows things to be different. On the other hand, asexual reproduction involves the process of what we call binary fission, which we'll talk a little bit more about that, what that means in a minute with bacteria. But it's where a bacteria divides from one cell into two cells. Or mitosis, same thing, going from one cell to two. This involves one parent, and the new cells look just like the parent cell. They are identically the same genetically. And so this allows for stability. It also allows for fast growth. We see that in humans, the formation of a new human is sexual reproduction, but the growth of the human or repair of the human, the cells will do asexual reproduction. All right, and so you have the two types of reproduction. All right, the next thing I want to talk to you about the characteristics of life. These are things that all living things have in common. So in order to be considered a living thing, you have to have these things. So for one, you have to be made out of cells. If you're not made out of one or more cells, you're not a living thing. Okay, so like viruses are not made out of cells. So they are considered acellular and non-living. We also see that this is going to be where metabolism comes into play. Okay, you have to be able to metabolize things. When we think of metabolism, a lot of times we think about how quickly you digest your food and the fact that you may not gain weight when you do so. But metabolism is more than that. Metabolism is all the chemical reactions that takes place in a cell or an organism. All right, and so this is where they build things or break things, but they are going to use chemicals and energy for growth and repair and all the different things in order for the organism to be successful. And so we call that metabolism. The next one is you have to be able to respond to your environment. This is where the organism has to be able to react, and the reaction is to help with survival. Okay, whatever that reaction may be. Now, this doesn't mean you necessarily run away or move away because plants don't have the ability to move like we do, but there may be some sort of response in order to have survival. The next one we see is reproduction. Organisms have to be able to do sexual or asexual reproduction in order to survive. And this is also to help perpetuate the species, meaning that we make more of them. All right, and so reproduction is an important part of living things. And last but not least is the ability to evolve and adapt. You have to be able to evolve and adapt to your environment. Otherwise, you don't survive either. All right, we talked about this. If you can't adapt and stay away from predators, then you get hunted down and you're not able then to pass on your genes to your offspring. Okay, so this is just to give some examples of things that all living things will have in common. All right, so now we have a chart that we're going to take a look at. This chart is going to look at the different kingdoms and domains that we're going to study in this class. And specifically 3 of these areas we're going to talk about in these notes. And then we're going to talk about the other two in different chapters. So let's take a look at this chart. This chart is found in your notes and you can fill it out. All right, so when we look here, we have the domains. The first group we want to look at is the bacteria and the archaea. These guys are going to be the prokaryotic cells. They're the ones, remember, that have no nucleus. They have no organelles, so they're very simple. There are two domains that fall into this category. There is archaea and there is bacteria. Okay, so those are the two that we're looking at. So they're prokaryotic cells. They're also considered unicellular. They have one-celled organisms. Now, if you'll notice, you're seeing a lot of these vocabulary words that we just talked about. So when we're looking at archaea in bacteria, we see that they can have individuals in the group that can do both photosynthesis or they are heterotrophic. Some are able to do what we call autotrophs and be able to do photosynthesis or chemiosynthesis. These guys are going to be what we call cyanobacteria, as well as the group for archaea. On the other hand, most bacteria are going to be heterotrophic and they have to actually consume their food. When it comes to motility, that's movement, it may vary. Some of these organisms are able to move and the cells have little tails that allow them to move called flagella. Others have these little structures called cilia that let them look like they're crawling across a surface, but they could have movement. Others, they may not. Okay, so it just varies. It depends on the organism. And last but not least, their life cycle is asexual reproduction. They reproduce asexually. So we are going to spend the rest of this particular set after we do our introduction talking about bacteria and archaea. The next video is going to focus in on protista. Protista is also found here in this chapter. These guys are now in the group or the domain called eukarya. Eukarya, remember, these are going to have eukaryotic cells. They have cells that have a nucleus and have organelles, so they're more complex. But guys, protista is the kingdom that's kind of like the junk drawer you have in your house. Stuff doesn't fit in any of the other categories, so it gets thrown into the protista group. So the protista group are unicellular with a few exceptions, and we do see that they get nutrition in 3 different ways. One is they could be these water and slime molds that are going to be heterotrophic like fungus is. They are going to act kind of like fungus and they're going to absorb their food. Okay, so they're going to liquefy it and they're going to absorb it. On the other hand, algae looks kind of like a plant. It is not a plant though. It looks like one. It kind of acts like one because it does photosynthesis. It's an autotroph, but it's not. And so it gets put in this category. The last group are the protozoas, and they act kind of like animals. They consume or eat their food like we do. The thing is that they miss a lot of the things required to be in the kingdom of Animalia. So again, they get thrown into this group called protista. The protista group is one that they're looking at with taxonomy, and it's probably going to change in the next few years, where it's not going to be the kingdom protista anymore. We also see with movement or motility, it varies. Some move and some don't. And also their life cycle. Some do asexual reproduction and some do sexual reproduction. We then see in the third video for this chapter, we're going to look at fungus or fungi. In this case, they are also eukaryotic. They are multicellular with a few exceptions like yeast. Yeast is a unicellular fungus. These guys are going to absorb their food and they are heterotrophic. They are non-motile, meaning they don't move on their own, and they do sexual reproduction. Okay, so they do sexual reproduction. Now they do this through a haploid type of cycle, which we'll talk more about when we get to that video. The next one is plantae. Plantae we're not going to talk about until another chapter where we're going to talk about plants. But plants are also eukaryotic, multicellular. They are autotrophic and do photosynthesis. They're non-motile, meaning they cannot move. And they do sexual reproduction through what we call alternation of generations. Again, we'll go into more detail when we get to that chapter. The last one is Animalia. Animalia are also eukaryotic. They're multicellular, but they're going to get their food from ingestion, bringing it in and being an ingester. They are motile, so all animals do move at some point in their life, and they do sexual reproduction, and it is a diploid type of reproduction. Again, we'll talk more about what that means when we get to those chapters. So a quick review here, we do have the 3 domains. You have Archaea, which are the prokaryotes that live in extreme environments like inside volcanoes, in hot springs, in Antarctica. They live in all those extreme places. We also have bacteria, which are the prokaryotes that are found everywhere. Okay, in you, on you, around you, in the air, the soil, the water, they're found everywhere. And then you have eukarya, which are all the eukaryotic cells. These include the kingdom Protista, fungi, plantae, and Animalia. All right, so let's review prokaryotes a little bit. Let's talk about some of their details about them. So prokaryotes, remember, do not have a nucleus and they do not have organelles, okay? The only thing that they do have are ribosomes. There are 2 domains, bacteria and archaea, and they are different because of molecular differences based on their DNA and all, and also some cellular differences, specifically their cell walls. So their cell walls are different. Now these guys used to be in the same kingdom together called Monera, but it has been separated due to our new knowledge from technology. As technology gets better and we get more knowledge, we have to make changes to taxonomy. These guys are found pretty much anywhere. They're found in soil, water, air, in you, on you. They're found pretty much everywhere. The most abundant of any organism is the bacteria group. If you were to take 1 gram of soil, like a small amount of soil, and you were actually able to measure the amount of bacteria in it, there would be 100 million bacteria cells in that 1 gram of soil. So they're everywhere, and there's a lot of them. Most of the time, they don't want to hurt you. Okay, these guys are really tiny organisms. They're very, very small. And there are more prokaryotes inside and on the human body than there are human cells in your body. So you have a lot more bacteria living in you and on you than you do just with your cells. Okay, so prokaryotic cells are found pretty much everywhere. So let's talk about domain Archaea first. So when we look at domain Archaea, they're going to have a composition of their cell wall, which is different than other types of bacteria cell walls. So this is part of the reason why they're put into their own group. They also have different types of ribosomes present. They still do the same job of making proteins, but they do look and act a little different. So again, this is why they have to be in a different group. They share more similarities actually with the domain Eukarya than they do with other types of bacteria. Their plasma membrane has a high fat or lipid content similar to ours. And most are autotrophic, but they do chemiosynthesis. Because they live in areas where sunlight's not present, they're going to be able to make their food from chemicals. These guys live in the really extreme environments. These environments are going to mimic what more primitive Earth looked like. So let's talk about some of these examples of the Archaea bacteria. The first one we want to talk about are the methanogens. The methanogens produce methane gas. These guys are found in your GI tract, and they're also found in other animals' GI tracts. They're also found in swamps and marshes, and they give that gas smell off. Okay, so they do live inside of us, and they are an example of one of the Archaea that do interact with us. The rest really don't. The next example are the haliophils. The haliophils survive in salty environments. Halio means salt, and phil means to love. So these guys love salt, and this is talking about 12 to 15% salt concentration. This is more salt than is found in the ocean. So they're found in things like the Great Salt Lake and the Dead Sea. Guys, the Dead Sea gets its name because if organisms wash into it, it is so salty that it sucks the water out of them and they die. They dehydrate due to the salt. And so these bacteria have made accommodations and adaptations to be able to live there, unlike anything else. We also see that there are the thermophils. The thermophils love temperature. These guys are going to love the hot temperatures. Some of them also love acid contents, and those might be acidophils. And sometimes they like the temperature and the acid, so we call them thermoacidophils because they like both. These guys are the ones found in hot springs or geysers like Old Faithful, found in Yellowstone, and even thermal vents down at the bottom of the ocean. Now, one thing to note about this group of Archaea is that none of them are pathogenic. So what does that word pathogenic mean? Pathogenic means that it makes you sick. Okay? If something is a pathogen, it's something that makes humans sick. In this particular case, none of these bacteria are known to make humans sick. Okay? So they are considered what we call non-pathogenic. The other group, when we talk about this, is the domain bacteria. They are also going to have a prokaryotic structure, and they are unicellular, but their cell wall is different biochemically than Archaea. Their cell wall is going to be made out of something called peptidoglycan. Okay, it's a kind of sugar protein mixture. Their plasma membrane also has a different fat composition than Archaea does. Again, this is why they're put in different groups, as well as their RNA looking significantly different. Archaea is more closely related to the bacteria, to the eukarya than bacteria. So again, why they are separated. Archaea also live in those more extreme environments. So it's important to note what the differences are between these because they do have similarities like being prokaryotic and unicellular, but they also have some differences. So continuing on with the domain bacteria, let's talk about this. These guys are the most common type of prokaryotes, okay, the cells with no nucleus. There are three basic shapes. Now, there are variations of these shapes when we talk about bacteria, but these are the three basic shapes, and you're going to take a look at these in lab. The first shape that we see here is coccus. Coccus has a round shape to it, and this should be easy to remember because if you look, a lot of the letters in the word coccus are all rounded. They have rounded edges. Okay, so they are the round shape. On the other hand, bacillus is the second one. So it's in the second picture here. And it is going to be rod shaped. So they're longer, okay, in their structure. These guys are easy to remember too, because if you look at bacillus, do you see the two L's? Those L's are showing you they're rod shaped. So bacillus is their rod shaped. The last one is really easy to remember because it's spirillum. Spirillum has a curved or spiral shape. Okay, so it's curved or spiral, and that's what you can see here in the last picture. Now, guys, it is important for you to know these bacterial shapes. You need to be able to match them to a picture. So pay close attention to this picture here. All right, so now let's take a look at the actual structures that are found in a prokaryotic cell, specifically for bacteria. All right, let's take a look at these. So we have a couple of structures that are going to be important in what we call the cytoplasm. Okay, the cytoplasm is the fluid that's inside of the cell. And in this fluid, you're going to find a nucleoid region. The nucleoid contains a single, what means one, circular, so it's a round strand of DNA. That is the nucleoid region of bacteria. Inside the cell, there can also be plasmids. Plasmids are smaller rings of DNA. Okay, these have actually been used by scientists for genetic engineering, where we can make bacteria make insulin for us or growth hormone, things like that, that we can package up and sell in pharmacies. Okay, but they're small little strands, circular strands of DNA. They're called plasmids. The last thing found in the cytoplasm are the ribosomes. The ribosomes are responsible for making protein. This is protein synthesis. The word synthesis means to make. So these guys are going to make proteins for the cell. The next set of structures are found in what we call the cell envelope. Okay, so an envelope protects a letter, like it protects it. It also hides it. It keeps it protected. So the envelope here is going to protect the bacteria. And the first part of this envelope is called the plasma membrane. The plasma membrane acts as a barrier between the inside of the cell and the outside of the cell. Okay, so it acts as a barrier, and it's going to determine what gets to come in and what gets to go out. It's kind of like the bouncer of a club. Okay, they get to stand there and decide who goes in, and if they have to kick people out. Okay, so the plasma membrane. The cell wall is outside of the plasma membrane. This is going to surround the plasma membrane, and it is composed of something called peptidoglycan. Okay, peptidoglycan. And if we break that down, peptio is talking about protein, glycan is talking about sugar. The last thing that might be present in this cell envelope is a capsule or slime layer. This surrounds the cell wall, and it's more of a protective type of covering. It gives it this idea of being kind of slick, and so it can stick to things or it can get away from things with this. Okay, so it's got a capsule or a slime layer. Capsules on medicine help us swallow them better. If you think about the medicines that are real chalky and they start dissolving when you actually try to swallow them, those guys don't have a capsule. But others have the capsule, which makes it easier to take and also makes it slide down in your throat easier. So these guys are going to help with protection, either a capsule or a slime layer. The last two on here are what we call appendages. These appendages are going to help the bacteria hold on to things, or they're going to help them move. So pili are short hair-like extensions, little hair-like extensions that are going to help them hold on to surfaces. Okay, so they're these little hairs that help them hold on, kind of like Velcro. If you've ever looked at Velcro and it sits there and kind of hooks on itself, okay, that's how these pili are going to kind of work. And we're going to look at a diagram in a minute that looks at all these structures. The last one is the flagellum. The flagellum is a whip-like projection that's off the cell, and it acts as a tail. And it moves back and forth, allowing the organism to swim or move in a particular direction. All right, so it acts as kind of like a tail or a rudder, and it's going to go back and forth, allowing it to move. Okay, so let's take a look at these on an actual diagram. All right, so if we look at this diagram, you can actually see that this has all the different layers we talked about. It has the cell membrane or the plasma membrane. That's the darker green on the inside. You then have a lighter green cell wall, and then the lightest one is going to be the capsule or the slime layer. Inside of the cell, you're going to see in the cytoplasm, there's the chromosome or the DNA. That's called the nucleoid region. The little sprinkles or dots are the ribosomes. Okay, and then if there were some little small circles, those would end up being the plasmids. You can also see the pili and the flagellum on here. All right, and so this is the basic structure of a prokaryotic cell. Now, the pictures right below this are what the plasma membrane and cell wall would kind of look like. All right, and so there's different structures on the cell wall on how they appear. The cell wall is all blue in the first picture, but then in the second, we've got a different layer where it's blue with then a fat layer on top. This is what gives different characteristics to bacteria as well, which also makes it harder for us to treat bacterial infections. That's why not all antibiotics work equally against different infections. And a lot of times it's because of the structure of the cell wall. The next thing we want to look at is the domain bacteria, is that there are some groups that kind of were misnamed as algae. In this area, there's cyanobacteria, and this used to be called blue-green algae, but it's not an algae, it's a bacteria. Now, this doesn't mean that it always appears blue or green. It can also appear red, yellow, or brown. These guys are going to be found in aquatic areas or very moist surfaces. They need water. Okay, so they can't be in dry areas like other bacteria could be. These guys require water. They also are going to do photosynthesis. So they're going to make their own food, and they're going to do this with the pigments found in thylakoids. Now, they do not actually have the organelle chloroplasts, but they do have the little poker chip-like structures called thylakoids that contain the pigments for photosynthesis, right? And so they are able to do that process. We do see that these guys are thought to release free oxygen into Earth's primitive atmosphere. So these guys are really important for oxygen production. Just like trees are important for oxygen production, these cyanobacteria are as well. These guys live singly, or they could live in colonies and makes kind of like filaments that are present. So if we look at this first one, these guys live more singly, like by themselves, unicellular. These are known as Glyocapsa, and they are going to live as single-celled organisms. If they live in a colony, this is Anabina. So they live in kind of a colony or group. And the last one that lives kind of in the filament form, like in a strand, kind of looks like a string, is Oscillatoria. These guys live in like what looks kind of like a little green string. And so these are all looking at them underneath the microscope because they are really small. But you can see that they are part of that group Cyanobacteria. Now, with cyanobacteria, they do lack the ability to move, so they have to live in water. Some of them do have a special structure called heterocysts that can help fix nitrogen. This is actually a good thing because we can't use nitrogen from the atmosphere, but plants and sometimes these guys can. So they have a special set of structures that let them do this. Anabina is a good example of one who has these heterocysts. We do see that these guys are the base of the food chain in water environments. So in water or aquatic environments, they're the bottom of the food chain. Okay, so they're kind of like how plants are the bottom of the food chain in other types of biomes that are on land. Okay, and so these guys are going to be the base of the food chain. So they are considered the producers. Now, we do think that these guys are thought to be where they colonized land, like moved onto land. And some of them are able to form what we call symbiotic relationships. Symbiotic relationships mean that they live with things. And this means that they could be good and beneficial with those things, or they could hurt them. An example of one where they actually work together and it's a good relationship where they are helping each other are when this bacteria works with fungus and they form something called lichens. These lichens are important in helping make actual soil. They grow on rocks and they break the rocks apart into soil. So this is a really beneficial type of relationship, symbiosis. On the other hand, there are times that these guys are bad for the environment. They can be associated with what we call pond or lake blooms, where they grow uncontrollably and they kill the fish. Okay, this means they have an overgrowth and the fish then die in that area. The next thing we want to talk about are biofilms. Biofilms are microbial communities that are held together in kind of like a gummy texture or matrix. And this again is a type of symbiotic relationship where they work together. It's called prokaryotic mutualism, where they're benefiting and helping each other. They also can make a really large structure that we end up calling a microbial mat. This is a large biofilm. It'll have lots of layers. These initially obtained energy from things like the hydrothermal vents at the bottom of the ocean. But now some of them can actually use sunlight as well. Okay, so it depends on their location, and they could potentially do photosynthesis as well. We know that these have been around for quite a while because these guys have been fossilized before. We have fossils of these biofilm or microbial maps that have been found. And these guys are called stromatolites. Okay, and so this is an example. Now guys, on your test, there is a short answer that you might have that says, what is a biofilm? What is a microbial mat? You have to be able to tell me. It also will ask, can they be fossilized? Yeah, they can. And so you want to make sure that you look at that for your potential short answer on the test. Another thing with these prokaryotic cells for bacteria is their reproduction is asexual. We see that they go from one cell to two, and they do this through a process called binary fission. So binary means that there's two. Fission means to split. So we go from one cell splitting into two cells. That's what binary fission means. This is found in prokaryotic cells where there is no nucleus. This is where one cell splits, which is the parent, into two daughter cells that are genetically the same. Okay, so they're genetically the same. So how does bacteria get to be different then? How come we have different strands of, say, E. coli? How do we have different strands of staph that cause staph infections? Well, DNA can vary in these guys because of three things. So let's talk about these three things. The first is called transformation. Bacteria is really good at incorporating new DNA it gets from the environment. So if it finds pieces of DNA outside of its body, it will actually pull it in and potentially use it. All right, and so bacteria is really good at doing that. The next way is through conjugation. Conjugation is where bacteria will share or transfer genetic information. So if one of them has some sort of gene that's beneficial, they can transfer it to another bacteria. And they do this by kind of touching pili and sharing that little plasmid, a little piece of the DNA with each other. The last way is transduction. Transduction is where we use viruses called bacteriophages, and we transfer DNA into a bacteria cell. This one is what we're going to use mostly in a lab setting, but we do see that they will then take in that DNA and use it. Now guys, these are also the ways though that bacteria has become antibiotic resistant, where it won't work against certain antibiotics anymore, is because of these three things: transformation, conjugation, and transduction. So these guys are the reason why that there are antibiotic resistant versions. Now, another thing that these bacteria could have to help them survive are called endospores. Now, endospores are formed by some bacteria when the environment becomes unfavorable or hostile, where normally it would kill the bacteria, but what the bacteria does is it encases itself in this kind of cocoon, and it waits. Okay, it doesn't grow, it just kind of goes into a dormant stage, and it can do this for years until conditions are better. The purpose of this is for survival and not reproduction. So when conditions become good again, or what we call friendly conditions, like food is back, the antibiotic has been removed, different things like that, these guys can break out of that shell and they can start growing again. Some of the best examples of these are anthrax and botulism. Anthrax, you may have heard about it. It was mailed to the Pentagon, to different types of senators and stuff back in the 90s. This is one reason why now they check all the mail before it goes to all those places. Anthrax is deadly. If you breathe it in, it can cause a bacterial infection in your lungs, which ultimately leads to death. Another one on here is botulism. Botulism creates a toxin that it releases that causes your muscles to be flaccid, meaning they don't work, they don't contract. We actually use the toxin from botulism to make Botox. Okay, so Botox takes that toxin and they inject it, and so that's why those muscles like in their face don't work. But they only don't work for a little bit. We don't give them the bacteria, we just give them the toxin. If we gave the bacteria, though, the patient could die. All right, so these guys are pretty deadly. They can be a big issue for us if we were to contract it. The problem is they can end up making these endospores and living for years, and we don't know that they're there. All right, some other bacteria that causes human diseases. And some have even been responsible for other pandemics. Now, we're not talking about COVID here because COVID was a virus, but we are going to talk about a few. So records as far back as 3000 BC show that bacteria have been a problem and have caused issues with the human population, causing sickness or illness. We also see that many pandemics have been caused by bacteria. Others have been caused by fungus or viruses, but bacteria have been some culprits. A lot of these bacteria actually travel from animals to humans. If that occurs, they're called zoonosis. They jump from an animal to a human, okay? And so this would be like a different kind of animal, like a pig or a monkey or something else, and they come and then affect us. Tuberculosis and the bubonic plague are two examples of pandemics that happened due to bacteria. Tuberculosis is a respiratory condition, and it is still common today. It actually kills 2 million people pretty much every year. You just don't hear about it that often. So it is still a big problem in the world. It is treatable now. You can't get rid of it completely, but you can live with treatment. On the other hand, bubonic plague is also known as the Black Death. This was carried by rats, and it then went to fleas, which then went to humans, and that's how humans contracted it. This killed a huge population of people back in the Dark Ages when we had the Black Plague, okay? And so these are just some examples. Now, an issue that we have and what can create more pandemics for us is the overuse and incorrect use of antibiotics. When antibiotics were first discovered, we thought they were like the magic pill and they cured everything. We thought that they could help us be able to overcome pretty much all illness. We were wrong. And because we used it for everything, we created a lot of bacteria that was antibiotic resistant. The other thing is when we have been given antibiotics, have you ever been one of those that didn't finish them because you felt better? Okay, don't do that anymore. Okay, always finish your antibiotics, because what happens is if you don't finish them, you leave behind the stronger ones. They may not make you sick again yet, but they are the stronger ones that when you take that antibiotic again, it won't work. Okay, so make sure that you take your full course of antibiotics if you are prescribed antibiotics. Another issue around here is that a lot of people go down to Mexico and buy their own antibiotics. Again, be careful of that, because not all antibiotics work against all bacteria. And if you're using the wrong ones, the wrong kind, it can actually make you sicker. So guys, natural selection keeps resistant bacteria around. They are able to survive, and this is why we haven't been able to kill out bacteria solely and all the way, okay? And so natural selection, like Darwin talked about, plays a role in this. However, MRSA is one of those examples where it has changed because of how we used antibiotics. Another group of bacteria that cause disease are the ones that cause food poisoning. These are the foodborne diseases or food poisoning from bacteria. So when you get food poisoning, you've gotten it because you consumed food that was contaminated with pathogenic bacteria, viruses, or other parasites. In the case of bacteria food poisoning, E. coli is one of the big culprits. And E. coli is a bacillus. It's a rod-shaped bacteria. And E. coli can be found in all kinds of stuff. We've seen it on recalls from spinach to peanut butter to canned goods. It can infect lots of different foods. And guys, E. coli is found in your intestines. As long as it stays there, it's great. But if it gets into other things, it can be very dangerous. Okay, it can cause a very severe food poisoning. Another example is botulism and salmonella. Salmonella is very common bacteria found in chicken. This is why it's so important for you to make sure that you cook your chicken all the way through, and whatever surfaces that you use and cut the chicken and stuff on, you clean them very well. Okay, so these guys are important. And we talked already a little bit about botulism, but botulism is going to be an issue if you don't can foods properly. Okay, that bacteria can get in there and it can cause problems and food poisoning if we don't can food properly. Some more examples of diseases. These guys are just bacterial examples for different types. We have STDs, which are sexually transmitted diseases like syphilis and gonorrhea. These are caused by bacteria. Respiratory diseases like strep throat or scarlet fever. These guys are caused by bacteria. Skin things like boils and pimples. In your digestive tract, you could have cholera or dysentery. Those are caused by bacteria issues. And guys, both cholera and dysentery cause the patient to have severe diarrhea and vomiting. So the patient becomes dehydrated very quickly. Some bacteria that can affect the nervous system are things like tetanus. Tetanus makes the muscles contract, and the contractions can actually be so great that they break the bones, okay, of the patient. All right, and so this is why if you've heard about how you can need a tetanus shot if you've been cut by something, this is why. Leprosy is a real disease. A lot of times you've maybe heard about it in like Bible classes or ancient books like that, where we talk about like the Bible and even the Quran and stuff. Leprosy is present. It's still present today. It's a bacterial infection that affects the nervous system, and it causes the tissue to die and pieces of the body just fall off as they die. All right. Leprosy is contagious. And so that's one reason why people who had it were put out of the communities and they lived in like what we call leper colonies. Systemic diseases, ones that affect the whole body, are things like typhoid fever. And then we also see Lyme disease. You may have heard of Lyme disease before, that's carried by ticks, but it is a bacterial disorder. So when we talk about all these bacteria diseases, a lot of times, some of them we don't die from, and this is because we can have treatments. There's also some that you don't hear about very often anymore because we can prevent them. So let's talk about treatment and prevention real quick. So with treatment, we do see that there are what we call antibiotics. These guys are going to work, they're medicines that work against the bacteria by either destroying the cell wall so your immune system can fight it, or by stopping the cell from doing protein synthesis so that your immune system can destroy it. All right. And so antibiotics can work one of two ways. And sometimes we have to pick different antibiotics based on how they work. Okay. Also, public awareness is super important when we talk about the spread of a lot of these diseases. So making sure that people understand good food prep techniques are important. Good hand washing techniques. That's one of the biggest things. We didn't think in 2020 we were going to have to teach people how to wash their hands again, but that's what we were doing. We've got to make sure that we have good hygiene there. And then also make sure we use antibiotics appropriately. This antibiotic crisis is a big deal. Another thing that helps with prevention is vector control. If some of these diseases are carried by fleas or ticks or other types of bugs, we can try to help control the population of those bugs, which then would help protect you from those diseases. Okay, so these are just some examples of how we could do prevention and treatment. All right, we only got a few slides left, and what we're looking at here for nutrition. What does this bacteria a lot of times need to do in order to get its food or nutrition? When we're talking about bacteria, they have to have some environmental conditions to help with their metabolism. They do the process of cellular respiration just like we do, but they may have to have some modifications. So one example is some of these guys are what we call obligate anaerobes. So let's break that down. If they are obligate, means they have to do it. Because if you're obligated, you have to do it. Anaerobes here means that there's no oxygen. So some bacteria have to live in places where there is no oxygen present. If oxygen's there, they would die. Okay. Others are facultative anaerobes, meaning they can live with oxygen or without oxygen. They're very versatile. They can do both. Oxygen's not toxic to them. They can live with it or without it. The last group are the aerobes. The aerobes require free oxygen to be present. They have to have oxygen. If oxygen's not present, they will die. Okay? Now, most of the bacteria that we grew in lab are going to be the ones who needed oxygen. Okay? Now, their mode of nutrition will also vary. Sometimes they can be autotrophs, like we saw with the cyanobacteria. Those guys are going to do photosynthesis. We also see the Archaea bacteria. Those guys are going to be able to do chemiosynthesis. But most bacteria are going to get their food from ingestion. These guys are heterotrophs. Okay, so most of the bacteria are going to be heterotrophs. Now, we talked briefly about some symbiotic relationships. I'm going to hit on them again here, but we are going to see these relationships come back over and over and over again. We're even going to do a lab on these and see how organisms work together. Now, sometimes when these organisms work together, we call it mutualism. Mutualism is where both the bacteria and its host are going to benefit. They're both going to get something good. This is like the nitrogen fixing bacteria that's found in soil and roots, and it helps the plant. Or the bacteria found in our intestine, which is going to help us with digestion, and we feed it. Those are actually good types of relationships, and we call them mutualism. The next one is called commensalism, and this one's a little harder to see, but commensalism is where the bacteria benefits, but you as the host doesn't get anything good or bad. This is what happens with some of the bacteria found on your skin. Okay, and you're going to see when you guys look at your plates that have the skin part, there's a lot of bacteria that live on them. That bacteria, you're helping it, but it's not hurting you or helping you. Okay, that's called commensalism. The last one here is parasitism. Parasitism is where the bacteria get something good, but you are harmed or hurt. The host is hurt. This is what causes diseases. These are the bacteria that make us sick, like the bacteria that causes strep throat or a staph infection or food poisoning. Those would be a parasitic relationship. The bacteria get something good, we are harmed or hurt. So we talked about kind of the bad stuff. Let's talk about some of the good things that bacteria can do, because a lot of bacteria is not bad. It actually helps us. And so let's talk about some of the economic importance. Now, economic means money. Okay? We actually see that bacteria is used to make a lot of things like alcohol, acetic acid, acetones, like the stuff that's going to be used on like your fingernails, and even antibiotics. So there's some good things that it can make. We also see that bacteria can be used to make food, like certain cheeses, like Swiss cheese, butter, sauerkraut. We have beers and wines as well as bread. Okay, sometimes those are going to be tasting different or made differently with different types of bacteria present. The other part is that bacteria is important in the ecosystem, the environment. This is because a lot of bacteria are what we call saprotrophs. Saprotrophs is just a fancy name for a decomposer. These are the bacteria who live off of dead material, so dead things. These guys are important for recycling nutrients in our ecosystems, and they also can be used in what we call bioremediation. We've used this for like pollutants, like when we've had oil spills, they have genetically engineered bacteria that can eat the oil. Our wastewater treatment plant here in Hobbs actually uses biomolecules, bacteria, to clean the water, okay, to get rid of the waste. So we can use it in that way for the ecosystem. Now, the last thing is not necessarily a good thing, but we need to be aware of it because of the fact that it could be a potential problem for us. Bacteria sometimes is used as biological weapons, meaning that bacteria could be released in order to cause massive chaos and illness in an area. You've all seen movies before where they are going to release some sort of virus or bacteria, and that's going to make a lot of people sick all at once and things like that. And they call it a kind of chemical or bio bomb, things like that. And so some of these bacteria are part of that we have to keep an eye on and we need to watch for. Anthrax is one of them. Botulism was another. We also see that it could be something like a tuberculosis that's going to be a respiratory disease. We have to be careful with this. I mean, we may not want to use it, but other countries might be thinking about it. And so we have to think about it too to offer us different types of protection. Because when we're not ready for things like that, that's when a lot of casualties and stuff may occur. Even though it's better to be ready and not need the information than not be ready for it. So these are just some examples. So this finishes up part 1 for bacteria. We are going to get into part 2 with protists in the next video, and then part 3 with fungus after that. If you have any questions, please let me know.